Let's start recording. I'm going to announce this in class and I'll put it up on the extra credit study guide. When you're applying for the extra credit study guide, you've got to be studying. A large number of people have been working during the lab time on the lab worksheet and then turning that in for extra credit. And in the future, I'm not going to give you any extra credit for that. So if there's any of the current lab, the current week's lab done on the extra credit assignment, you will get a minus a half a point for each lab. So the most you'll get is -1. And since you get 1 point of extra credit, it'll be 0 points. But the point is, I don't want you working on the lab worksheet for the extra credit study. For the extra credit study, I want you guys studying. Any questions about that?
Yeah, can you explain what you want us to study, since we considered studying the lab together after the lab Zoom call, that would count?
I don't care if you study an old lab for the quiz. However, the current lab is an online lab and you have to do the worksheet. So I'm not going to accept that for studying. Okay. Normally in the lab, you go into a lab room and you do the lab work. This is an online lab, so obviously we don't have that. I'm not going to give you extra credit for attending the lab. Is that clear?
So then would we study like for the extra credit, like I know we have the study guides that
Study
you
the
provide.
material for your quizzes.
Okay.
Okay. And you can study other material. I'm not gonna, I mean, it has to be microbiology, but,
Right.
you could be studying for your final. That's going to be comprehensive. So,
But that could be the extra credit.
what?
But that would be counted as the extra credit. That's acceptable.
Yeah, I'm just saying I've never had students who worked on the current lab and then submitted that for the extra credit study. That's not studying. That is your
No, I
lab work.
understand.
Okay, you are supposed to be in lab. And it's true, we have an online lab because we're an online class. And
We
you
attended the
can work
lab. We
together.
studied after.
I'm not going to say you can't work together. Okay, for the lab. But in that week, I'm not going to accept that week of the lab for extra credit. Okay. All right. Now, I think I will accept everything that's been submitted up until last, I don't know, what was that, Sunday? So you will get your extra credit, but I was really disappointed. About a third or maybe even half the class is doing that. And about a third of them, that's all they're doing for the extra credit studying. And that is not studying. Okay, I want you guys quizzing each other on the material you're going to need to know for the quizzes and the final. Now, you don't have to quiz each other. You can study in other ways, but all right, I've stated that and that's done. Let's move on. Are there any other questions? I should probably plug that in. I don't think you can see my screen yet, but that'll be changing shortly. Sharing the screen now. I thought I opened up the, oh, there it is. It's just taking a while to open. Come on. So today is October 12th, and we are supposed to be on chapter 4. I think we did finish chapter 3, didn't we? No comments? Okay, well, we'll start chapter 4 today. Please note, on Wednesday, you do have quiz 2. It'll be covering labs 3 and 4 and chapter 3 and the end of chapter 2, only the end of chapter 2. Any question about any of that? All right, we'll talk about the lab. In the lab time today, we do only have a lab on Tuesday this week. All right, let's move on.
Do we have lecture on Thursday?
Yes,
Just not lab. Okay, thank you.
yeah. Well, there is going to be a question and answer session if you want to ask questions on Thursday. Let me see, I'm going to keep that up. And chapter 4. Come on. I'm not seeing the icon here. Boy, I lost control. There it is. Okay, there's the icon. So I am recording. I just want to make sure of that. There it is. All right, chapter 4, functional anatomy of prokaryotic and eukaryotic cells. If we do not finish this chapter in time, I will assign the eukaryotic cell part of the chapter to you guys to finish. You can read it and I'll give you the slides, but you should have had that in your prerequisite course. So that should be totally review for you. All right, if you're coming in late, you need to hear the announcement I made at the start of the assignment or the lesson. So this slide is showing you the major goals and the rough outline. Know the terms of this chapter. The different shapes of the bacteria, coccus, bacillus, spirilla, the different growth arrangement like streptococcus, staphylococcus, diplococcus, tetrad, sarxinia, vibrio, spirochete, and then know the parts of the cells, the glycocalyx, also called glycocalyx, both pronunciations are correct, flagella, axial filaments, fimbriae, villi, peptidoglycan, endotoxin, selective permeability. Well, I guess that's not a part of the cell. Well, it's a little more than part of the cell. It's a concept. Diffusion, facilitated diffusion, osmosis, active transport, tonicity, what's the plasmid, what are inclusions, endospores, and the endosymbiotic theory. We will cover the endosymbiotic theory even if we don't finish chapter 4, meaning I will cover that topic. You should know how gram-positive and gram-negative cell walls differ and describe the importance of taxis in bacteria. When we're looking at the prokaryotes and comparing them to eukaryotes, you should know that prokaryote are Greek words, pro and karyote, for meaning pre-nucleus. Literally, it means pre-kernel, but we'll take that to be pre-nucleus. And then eukaryote is Greek for true nucleus. Any question about that? The prokaryotes generally have one circular chromosome, meaning it's a circle, it ends where it begins, and the chromosome is not in the nucleus. Eukaryotes, on the other hand, have a linear chromosome, like the X chromosome, has the beginning point and an ending point. and their chromosomes are in the nucleus. When we look further at the DNA of the prokaryotes are not associated with histones. Histones are proteins found in eukaryotes, and in eukaryotes, the DNA wraps around the histone protein. So there's a lot of histone protein in the DNA, in the chromosome of eukaryotes. And when we say chromosome, you should understand that means both DNA and its associated proteins. In eukaryotes, the number one protein is, of course, histone proteins, but prokaryotes also have DNA in their chromosome. It's just not a histone protein. Any question about that? Prokaryotes don't have any organelles. By organelle, I mean a membrane-bound organelle. And eukaryotes do. They have membrane-bound organelles, like the mitochondria, the chloroplast, the Golgi complex. We find peptidoglycan in bacteria only, in their cell walls. And the eukaryotes, we do not find peptidoglycan in their cell walls. Of course, animal cells don't even have cell walls. Prokaryotes, their cells mainly divide by binary fission. We'll talk more about that, but that's where the cell literally divides into two. And eukaryotes don't do that. They divide by mitosis. Any question about that? All right. So we're going to talk about the prokaryotes, talking about their cell shape and their cellular growth arrangement. We'll then move into cell structures, the movement of material across membranes. And if we have time, we'll talk about the eukaryotes, their cell structures. And then we will cover the last part of this lesson, and we will talk about the endosymbiotic theory. Prokaryotes have a cell size and shape. Their average size, this one is saying 1 to 10 micrometers in length. 10 would be a very large prokaryotic cell. Most prokaryotes are between 1 and 2, or maybe 1 and 3 micrometers. There are 3 basic shapes to prokaryotic cells. They can be round, and we call those coccus, right here. Bacillus, there can be rod-shaped, and those we call bacillus. And these two terms are used interchangeably in microbiology. So if you call it a rod, that's not incorrect. If you call it a bacillus, that's also not incorrect. If you call it a coccus, that is the correct term. You should not be calling it a round cell. Okay, any question about that? And then the third basic shape is a spiral, has an S-type shape shown here. There are different types of spirals. I wouldn't even call this a type of spiral. I would call it a rod that's bent. But a vibrio is a bent rod, a curved rod. Okay. Any question about that? They're saying it's a half of a spiral. That's what they're saying.
If we were being questioned on the vibrio, would we be marked wrong if we answered that as a curved rod, or would you want us to answer that that was a type of spiral?
Well, that's what I'm saying. I won't ask that question, but in my opinion, it's a curved rod, but you could say it's a half of a spiral.
Okay.
Okay. The spiral, we've already talked about the main shape. And then there's a spirochete. It's a spiral with many, bends in it. And there's a little bit of a difference in the spiral and the spirochete. And that is the spiral is rigid. The spirochete is flexible. You can bend its shape easily. All right, I'll talk about these two in a little bit more detail because they're a little bit unique, and not this slide, but the next slide. Prokaryotes can have some unusual shapes. You don't need to know these, but there's the star-shaped Stella bacteria that actually look like this. That's an actual electron microscopic picture of them. A little bit less than a star. Well, that one has 5 points to it. And then there's the square, actually I would say rectangular, Haloarcula. That's an archaea. Most bacteria species are monomorphic, meaning they have one shape. You look at them under the microscope, they'd all be rectangular, they'd all be star-shaped, they'd all be rod-shaped. But there are a few species which are pleomorphic, meaning they have many different shapes. Now here's a picture of a pleomorphic bacteria. Most of the pleomorphic bacteria, you don't need to know this, but they belong in one family, the Mycoplasma, and they're pleomorphic because they don't have a cell wall to give the bacteria a shape. So there actually is one family of bacteria which do not have a cell wall. And if you remember when I was talking about peptidoglycan, I always worded it as you find peptidoglycan in the cell wall of bacteria. Well, the mycoplasmas don't have a cell wall, so they don't have peptidoglycan. But the point is, there are a few species which are pleomorphic, and you should know that term. We really won't talk about the mycoplasmas, so you don't need to know that. The prokaryotes can be described as having a cellular arrangement or a cellular growth arrangement. That is how the cells stick together after they divide. Is that clear? So we have one cell that can then divide. If the cells stick together, we get a cellular growth arrangement. And that's how the cells grow or stick together. You can have singlets. That's not up here, although it's right there. That's a singlet. So you can have singlets. And there are a few species which you will only see singlets. That's usually the spirals or the spirochetes. We only have singlets. But other species can have other growth arrangements, such as a diplococci, where two cocci stick together. And I think you might be familiar that there are some human diseases which are diplococci. There's also a streptococci, and I'm sure you all know about strep throat. That's a streptococci, where the bacteria grow in a chain in their cocci. There are also bacteria that form a tetrad, and then bacteria which form a Sarciniae, that's 8, a group of 8 cells. And then there's bacteria that form a cluster like grapes. And Staphylococci actually means cocci in the appearance of a cluster of grapes. So that's what staphylo means, cluster of grapes. And why they do this is just the way they divide. You don't really need to know that, but if they grow only on this plane, they'll either form diplococci or streptococci. And if they grow on 2 planes, they can form a tetrad. On 3 planes, they can form a Sarciniae or a staphylococci. Now, the cocci have more cellular growth arrangements than the bacilli. With the bacilli, you can have single bacillus, so that's a growth arrangement. You can have diplobacilli, 2 of them. You can have streptobacilli, a chain where they're attached on the long end. And you can also have a palisade, the chain where they're attached on the shallow width. And if you flip that sideways, you can't do that, but they're sort of like this. And if you flip it that way, it actually looks like a palisade fence. which if you read Mark Twain, they talked about a palisade fence in Mark Twain. It used to be very common. You just took a board and then you stuck it in the ground and attached it to the next fence. And I guess you nailed it. I don't know what you did, to tell you the truth. I never made a palisade fence, but you just put those in and there you had your palisade fence. Anyways, they thought those looked like a palisade fence, so they called that arrangement a palisade. Any question about that? Now, this is not a growth arrangement, so I don't know why it's on this slide. The Coccobacillus is a different type of cell. And I mentioned that there are different shapes. Well, there's a shape where you get a bacillus and you get it smaller and smaller. If it's rounded on the ends and you get it small like this, it's in between the shape of a coccus and a bacillus. And that we call a coccobacillus. Any question about that? All right, so don't confuse the shape of a cell with the families. There is a family of bacillus, and they happen to be all rods, so they do have bacilli in them. But if it's a shape, it should be a lower B. If it's the family, it should be a capital B. And that's how you can tell the shape from the family. Any question about that? And the same with Streptococcus, that is the family. Streptococci is the cellular growth arrangement. And usually this would be a lowercase s for Streptococci. Any questions about that? All right. Anything else I want to say about that? So the bacilli have fewer growth arrangements than the cocci. And then the spirochetes and the spirals only have one growth arrangement, and that is a singlet, a single cell. They don't stick together for whatever reason. When you see a cellular growth arrangement under the microscope, you will not see one cellular growth arrangement. You will see a mixture of them under the microscope. So if it's a streptococci, it can break off right here, and then you'll have diplococci. So you can see some diplococci in the field of view along with the streptococci. And then if this can break off, you'll have a singlet. So usually with the streptococci, you see 3 different cellular growth arrangements. When we name the growth arrangement, you take the highest order. So that would be a streptococci. Any question about that? All right. Some different structures of a typical prokaryotic cell is shown here. Let me blow this up. We'll talk about each of them, but there are 3 structures that you can find in all cells. All cells have the cytoplasm, all cells have the plasma membrane, and all cells have a DNA. So those 3 structures you'll find in all cells. Now it's true that all cells have ribosomes, but the ribosomes of the prokaryotic cell is different than the ribosome of the eukaryotic cell. They largely differ in size. Any questions about any of that? All right. Remember when we're talking about prokaryotic cells, they have a nucleoid region. This is the region where the DNA is floating in the cytoplasm. wherever the DNA moves to, floats it to, that will be the nucleoid region. There is no distinction between the nucleoid region and the cytoplasm because there's no boundary in a prokaryotic cell between the two. It's wherever the DNA floats in. And the DNA typically stains differently than the cytoplasm. And that's why they call it the nucleoid region. Any question about that? All right. These other structures are found in, well, probably most prokaryotes, but not all prokaryotes, with the exception of the capsule. That's found in, well, I suppose also the, never mind. The capsule, the inclusion, and the pilus are not found in most cells, most prokaryotic cells. Okay. The cell wall is found in most prokaryotic cells. We already mentioned the plasma membrane, also called the cell membrane. That's found in all prokaryotic cells as well as eukaryotic cells. The fimbriae, found probably in most prokaryotic cell. I'm not 100% certain of that, but certainly many prokaryotic cells have fimbriae. Flagella, actually that one is not found in most prokaryotic cells, but many of them have flagella. Remember, if the bacteria or the archaea, if they swim, move by swimming, that means they have to have a flagella, because that's the only structure that a prokaryotic cell has for swimming or moving. In prokaryotic cells, you can find the DNA in the chromosome, as well as in a small region of DNA or small molecule of DNA that we call a plasmid. All right, any questions about any of this slide? The glycocalyx, also pronounced glycocalyx, is secreted by many prokaryotes, but not all prokaryotes. So the majority do not have a glycocalyx. It's outside the cell wall. It's usually sticky. And there are 3 types of glycocalyxes. If we can see it under the microscope, a light microscope, then that type of a glycocalyx is a capsule. It's a neatly organized structure outside the cell wall, and it can prevent phagocytosis. That's where a white blood cell comes up to a bacteria and then engulfs it to eat it and then kill it. The capsule can prevent that. If the glycocalyx is unorganized, largely water, and loose, we call it a slime layer. A slime layer cannot be seen under the light microscope. You would need an electron microscope to see the slime layer. There's a 3rd type of a glycocalyx, and it's only found in biofilms, an extracellular polysaccharide that the bacteria secretes external to itself. And that we call the extracellular polysaccharide, or EPS, which is only seen in biofilms. We'll talk briefly about biofilms later in the term. All right, any question about this? So if you can see the glycocalyx under the light microscope, it's a capsule. We even have a stain for that, as we mentioned last time. The flagella is largely outside the cell, but there is a portion of it attached to the cell wall and attached to the plasma membrane. So not all of it is outside the cell wall. It's a whip-like structure, and it beats to propel the bacteria to move or swim through the water. So the flagella is used for locomotion, It's largely, but not entirely, outside the cell wall. And it's made of chains of flagellin. Flagellin is a protein. I won't ask you the different parts of the flagella. I think we already studied this in the lab, and that is the flagella can be in 4 basic arrangements. It can be one on one side of the cell, and we call that monotrichous. There can be 2 on each side of the cell, and that's amphitrichous. There can be multiple on both sides of the cell, and that we call lophotrichous. And then there can be many all over the cell, and that we call peritrichous. As I mentioned in the lab, when you see the cells under the microscope, assuming they don't have a monotrichous shape, monotrichous bacterial flagella arrangement, they will be more than one, meaning a peritrichous. When it first starts to form, it'll be monotrichous. And then as it gets more developed, it'll be amphitrichous. And then it'll become lophotrichous. And then eventually it'll become peritrichous. So when we look at them under the microscope, you need to name it, which form it is, by its highest flagellar arrangement. Any questions about that? And I think some of you got that wrong in the lab because you looked at one of them and it was this or that, and you said it was that. And that one you were looking at was, but if you look around the slide, there were peritrichous in it.
Thank
Do
you for
not
explaining
memorize-
that.
I'm sorry?
Thank you for explaining that. I am one of the people that got that wrong.
Okay. I thought I mentioned that in the lab last time, but that's all right. Do not memorize these names. I'm not going to test you on it. You just need to know it for the lab worksheet and maybe the worksheet in the lecture today. I don't remember. But I'm not going to test you on the different names for the bacterial flagella. So if the bacteria cell is motile, it has flagella, and the cells can rotate or run or tumble. And this is a swimming-like motion. This allows for taxis, the movement toward or away from a stimuli. That's what taxis is. In this case, there's a light source up here, and the cells are moving this way, away from the light source. That's actually rare. Most bacteria that have phototaxis, meaning they move in relationship to the light, they will move towards the light because they want probably to engage in photosynthesis, but they want the light. Most bacteria cells do not move away from the light. Anyways, there's 2 types of taxis. Phototaxis, where the cell responds to or against, moves towards or away from the light. And then chemotaxis, where the bacteria is responding to some chemical. And they'll either move towards the chemical, like if it's glucose, or they'll move away from it, some toxic chemical. Any question about any of that? All right. Now here's one that doesn't usually cause too much trouble for students, and that is you can have a flagella which is special, and we can call it an axial filament, and I don't use that term. I tend to call it an endoflagella. It is a flagella inside the cell. You find the endoflagella only in spirochetes. It's anchored at one side of the cell, this side or that side. And then it wraps around the cell. And when it moves, it gives the cell a corkscrew-like motion. don't know if I can do that. We'll take a look at it, corkscrew type motion. But note that the endoflagella is inside the cell wall. Let me blow this up. So you can see it wrapping around the cell, and there it's an artist's rendition wrapping around the cell. But when we cut it this way and then look at it, there, blow it up a little further. The pink is the cell wall, and you'll notice the axial filament is inside the cell wall. It has pink outside the endoflagella. So this flagella is totally inside the cell because the cell wall is the last portion of the cell that we consider to be part of the cell. Anything that's external to the cell is, well, and anything that's external to the cell wall, we consider to be external to the cell. And I forgot to mention that when I was talking about the capsule, it's secreted by the cell and it's external to the cell wall. So we consider that outside the cell. Any questions about any of that? The endoflagella is inside the cell. Shrink that down to a regular size. Let's go and take a look at the video. Why didn't that work? Yeah, I'm sure you want to see the commercial.
$1 billion stimulus program. It's a brand new stimulus program that will literally pay you to.
So as you can see, not only can it move back and forth, but it can twist. The spirochetes are flexible and it bent and then changed its direction. And that's how it's moving in a spiral corkscrew type movement. And it literally swims through the media that way. Start that over again and move over here. So right there, see how it just switched? It was going up and down, now it's moving back and forth. What would I want to say about this? Oh, yes. It's believed that the spirochetes, because of their corkscrew type movement, can actually burrow through human tissue. So if you have a spirochete, which is a pathogen, it can actually burrow through human tissue, including the mucous membrane. And that is why spirochetes are a, I don't know, contagious disease. They can burrow through the mucous membrane and attack mucous membrane. All right, any questions about the endoflagella? The fimbriae are shorter and straighter than flagella, and they're used for attaching to a surface. They're not used for swimming. Any question about that? And they are yellow structures here. Similar to the fimbriae is the sex pili, but it is longer and a little thicker and it's hollow. The sex pili is used to transfer DNA from this cell, one cell, to another cell. And the DNA can actually move through the hollow pili and move from this cell to that cell. This sex pili does act as a winch, and so it can bring this cell really close to this cell. So it does literally go out and skewer another cell, and it can, if it wants, bring it in closer. So it acts like a winch to bring the two together. But they don't need to be together because the sex pili is hollow. The plasmid can move from this cell through the sex pilus into that cell. All right, any question about the sex pilus? By the way, the plural is pili. Pilus is singular. All right, let's talk about the prokaryotic cell wall. It's a complex semi-rigid structure responsible for the cell shape of the prokaryotic cell. It does protect the cell and prevents osmotic lysis. If you were to put a prokaryotic cell with a cell wall in a beaker of water, water would move into the cell and the cell membrane would expand, but it could only expand up into the cell wall. After that, the cell membrane can't expand any further. And if the cell membrane can't expand any further, no more water will enter the cell. On the other hand, if the bacteria lacks a cell wall, or we're talking about a red blood cell, which doesn't have a cell wall, and you put it in a beaker of water, water will move from outside the cell to inside the cell, expanding the cell membrane, and keep on expanding the cell membrane until the cell membrane will eventually burst. And then that'll leak the cytoplasm out of the cell, and that'll kill the cell. So the cell wall can prevent osmotic lysis, protecting the cell. The cell wall is located outside the cell membrane. In some species, the cell wall can contribute to the pathogenicity. We'll talk briefly about the cell wall of Mycobacterium. That cell wall can contribute to the pathogenicity of that bacteria. The cell wall is largely made up of peptidoglycan. We're talking about the bacteria, not all prokaryotic cells now, because the archaea essentially don't have peptidoglycan. But the bacteria do, and it's the main molecule in the cell wall. For the gram-positive cells, it is vastly the main molecule, meaning like 90 to 95% of the cell wall is made up of peptidoglycan, and the other remaining is other molecules. All right, any question about peptidoglycan? If not, let me see, where's the best place to put that? I'm going to put it there. Peptidoglycan is a polymer of a repeating disaccharide. The two disaccharides which repeat are NAG and NAM. NAG stands for N-acetylglucosamine, and NAM for N-acetylmuramic acid. I'm sure NAG and NAM mean as much to you as N-acetylglucosamine. Anyways, peptidoglycan, all it is a chain of NAM and NAG, repeating NAM and NAG. And then that chain has parallel chains in the plane. And then the plane has parallel planes, in this case, underneath it. If we were this one, it would be above and below. The chains are cross-linked with short peptides. So short peptides link the chains together. And usually there's two, and that is this one runs from, well, let's do this one. This one runs from this peptide to that peptide. This peptide is holding that NAG and NAM chain together. And then this one down here is holding onto this chain of NAG and NAM. And then this one is doing the same. It's holding these two together. So this peptide is holding this to that indirectly, meaning it's linked to this short peptide and this short peptide. Your artist of your textbook diagrams the NAG and NAM this way, and then this way, meaning there's more than one. And I prefer this picture because it does show you in detail much more how NAG and NAM are and the relationship with the short peptides. Now, what did I want to say? Oh, yes. NAG and NAM are disaccharides. And saccharides or sugars are called glycans. And then there are peptides, short peptides, linking the glycans together. And that's how we get the name peptidoglycan. Glycan with peptides, peptidoglycan. Any question about that? Anyways, the peptidoglycan forms a lattice which encompasses the cell, giving the cell its cell wall. Penicillin actually inhibits the lattice formation when the cell is putting down peptidoglycan, and we'll talk more about penicillin later in the term. But if you give penicillin, I'm talking about penicillin G, but it could be any member of the penicillin family, it'll stop the peptidoglycan from forming, and that'll create holes in the cell wall. Hopefully you can see that hole there. And the cell membrane will protrude out of that hole. And so water can move into the cell membrane will expand. It'll expand out of the hole right here because the peptidoglycan is being blocked by penicillin. And as more water moves into the cell membrane will expand further out of this hole. And eventually it'll expand so far that the cell membrane will pop or burst, and then that'll kill the cell. And that is how penicillin kills bacteria. They prevent the cell wall from forming, and then water moves into the cell, expanding the cell membrane. The cell membrane comes out the hole in the cell wall, and it'll eventually pop. Anyways, that's how penicillin inhibits the growth of bacteria. Any questions? All right, so we've already talked that there are two types of bacteria in relationship to their bacterial cell walls. There's the gram-positive bacteria shown here. Usually, well, it doesn't have to be a rod. The purple is the peptidoglycan, and you see that's very thick. The yellow is the lipid bilayer of the cell membrane. And then we're looking at it blown up here. You'll note that there's many layers of peptidoglycan. The artist's rendition is only showing you 3 layers. It's many, many times more than 3 layers of peptidoglycan in the gram-positive cell. It's hundreds of layers of peptidoglycan in the gram-positive cell. There are a few other molecules in the cell wall, but the vast majority of the molecules are peptidoglycan. The cell wall is attached to the cell membrane, and that's how the two stick together. There are other molecules in the gram-positive cells. There's teichoic acid, And it's a molecule that runs lengthwise through the peptidoglycan. There's also lipoteichoic acid. It has a lipid on the teichoic acid. And this molecule does run through the peptidoglycan, but the lipid portion runs into the plasma membrane. And that's why it's got the lipid portion on it. The lipid portion can bind to and be in the plasma membrane. Teichoic acid may help regulate the movement of cations. Cations can move through the cell wall, and it's believed that they can use the teichoic acid to move down the teichoic acid so that the cation can move towards the cell membrane, then come into the cell. Anything else I want to say about that? There are polysaccharides in the cell wall, and they can provide antigenic variation, which humans can use to differentiate among two different species. Like E. coli, the normal one, I don't even know what its number is because nobody ever calls it E. coli such and such. This is the normal one. It doesn't have toxins, but E. coli 0157 H111 has carbohydrates on it that give it the numerical designation I told you. What did I say? 0157 and H111. Those are polysaccharides in that E. coli, which can change the behavior of those cells. And E. coli is not normally toxic to people, assuming they don't consume a very large number of them. Even if you were to eat E. coli, if you ate too much of it, it would give you slight health problems, mainly diarrhea or constipation. Well, E. coli 0157 H111, the polysaccharides are toxic, and these cause real gastrointestinal irritation. Any question about any of that? All right, the gram-negative bacteria cell wall differs. And first, when we look at it, we'll note that it does have peptidoglycan, but it's a very thin layer comparing to the gram-positive cells. And then it has the lipid underneath, which is the same. That's the cell membrane. And then it has a lipid bilayer above it. And that is the outer membrane of the gram-negative cell. So let's blow this up. We have in the gram-negative cells, of course, the cell membrane, which is a lipid bilayer. We do have peptidoglycan also. And please understand this artist is drawing it with only one layer. In reality, it's at least a few layers of peptidoglycan, and it might be many, but it's many layers less than the gram-positive cells. Any question about that? All right, no questions, let's move on. In the bacterial cell wall of the gram-negative, we have other molecules besides peptidoglycan. And if you look right here, this is a lipid bilayer called the outer membrane of the cell wall. And the lower bilayer is the same molecule that we see in the cell membrane. And that is, it is a phospholipid. Now, in the upper part of the bilayer, or the outer part, if you want to call it that way, we do have phospholipids, those yellow molecules with the tails on them. So that is found in the outer lipid bilayer of the outer membrane. But we find another lipid, lipid A, come on, mouse, right there, or right there. And it is a lipid, so it stays in the lipid bilayer. And there's usually an O polysaccharide attached to lipid A. So this molecule is a lipopolysaccharide, meaning a lipid and a saccharide, carbohydrate. A lipid A is the important molecule because lipid A is an endotoxin. We'll talk more about toxins later in the class, not today, but toxins are something that are toxic to people. And so getting them is usually a bad thing. Well, lipid A is found in the outer membrane of the gram-negative cell. So if any patient has an infection by a gram-negative bacteria, if the cell has a cell wall, it will contain lipid A. And lipid A is an endotoxin. Any question about any of that? All right, we find a few other molecules in the gram-negative bacterial cell wall. There is a porin protein, and it is, well, it sort of acts like a transport protein, and that moves molecules that the cell wants inside the cell through the outer membrane. And then molecules that the cell wants outside of the cell, the porin protein can also allow that molecule to leave the cell. Now that's important because remember, this is a lipid bilayer, the outer membrane. And so many molecules cannot get through it. And how they get into the cell or out of the cell is through this porin protein. Now realize that porin proteins are what's the word I'm looking for? They're not a transport protein, because a transport protein generally only works on one molecule and then either moves it into the cell or moves it out of the cell. The porin protein works on many different molecules, and many different molecules can move through this protein channel. That's the word I was looking for. And that is, the porin protein is a protein channel, allowing many molecules to move through the outer membrane of the cell wall, as well as allowing them to leave the cell, many different molecules that would not get through the lipid bilayer. If you don't know, and I guess we haven't talked about that, most water-soluble molecules, meaning most polar molecules and ionic molecules, will not get through the outer membrane or the cell membrane unless they have a protein to bring it through that membrane. Any question about any of that? All right. So the porin protein differs from a transport protein in that a transport protein usually only allows one molecule to either enter the membrane or to leave the membrane. The porin protein allows many different molecules to enter or leave the outer membrane of the cell wall. I think we talked about everything except maybe we can find lipoproteins and, well, we already mentioned phospholipids in the outer membrane of the cell wall. So it's not just phospholipid. There are other molecules. You can also find proteins in the outer membrane, like the porin protein is a protein in the outer membrane of the cell wall. I think I've already talked about everything here. I'll word this, and that is the boron protein allows many molecules to enter the outer membrane, such as glucose, fructose, amino acids like cysteine, methionine. Okay. But you need a transport protein to move the molecules through the cell membrane. And so for each molecule you want to move in, you'll need a separate transport protein, meaning there's one transport protein for glucose, there's one transport protein for, well, I assume there's one for methionine and one for cysteine. I assume that, but I actually don't know because I've never read it. But the cell would want those molecules inside the cell. So I'm going to bet that there will be a transport protein to move that molecule into the cell. All right, so comparing the gram-negative outer membrane, we see that they have lipopolysaccharides, lipoproteins, and phospholipids. I'm not going to quiz you on the periplasm. That is the space between the outer membrane and the plasma membrane. That, I would say, that's where the peptidoglycan is, in the periplasm. So I just call it, that's where the peptidoglycan is. The gram-negative outer membrane can help protect the bacteria from the complement proteins and from antibiotics. As I mentioned, penicillin G cannot penetrate the outer membrane of a gram-negative cell. So penicillin G will not work on a gram-negative cell. It can work on a gram-positive cell. The gram-negative membrane can also protect the cell from the complement proteins, already mentioned, from certain antibiotics. It can help prevent phagocytosis. And as I mentioned, there could be a saccharide on the outer membrane, which gives us antigenic differences. And here's one like E. coli. We're not just calling it E. coli. It's of the polysaccharide O157H7. And I think I've got the O polysaccharide. Yep. This is the O polysaccharide. It is the saccharide attached to lipid A in the lipopolysaccharide. So that's the O polysaccharide. I already mentioned that. All right. Let's compare the gram-positive cells with the gram-negative cells. Gram-positive cells have thick peptidoglycan. The gram-negative cells have thin peptidoglycan. There's teichoic acid in the gram-positive cell walls. There's no teichoic acid in the gram-negative cell walls. There's no outer membrane in the gram-positive cells. There is an outer membrane, and it contains lipid A in it in the gram-negative cells. Any question about any of that? All right, the cell walls give rise to the gram stain. The crystal violet iodine complex forms in the cell, specifically in the cell wall. The gram-positive, the crystal violet iodine crystals do not leave the cell because of its thick peptidoglycan. It doesn't wash off. In the gram-negative cells, the alcohol dissolves the outer membrane of the gram-negative cell, and then the crystal violet washes out of the thin peptidoglycan. In the gram stain, we then stain with safranin so we can better see the cells. So safranin stains the unstained cells pink. This slide is showing you a summary of gram-positive and gram-negative. You only need to know the top portion. You can ignore the lower portion. We already talked about that. Teichoic acid we mentioned is present in the gram-positives, absent in gram-negatives. Periplasmic space absent in the gram-positives, present in the gram-negatives. The outer membrane, absent in gram-positives, present in gram-negatives. And then the lipopolysaccharide layer containing the endotoxin, lipid A, virtually none in the gram-positive cells, and a high number of this toxin in gram-negative cells. All right, any question about any of this? If not, let me briefly state that there are some cells which are odd and different from the cells I've shown you, meaning I've talked about them for the gram-positive and the gram-negative cells. The first one I'm going to talk about is archaea. They have cell walls not of peptidoglycan, but of pseudomurine. That's another disaccharide. And there are a few archaea that even lack cell walls. So the archaea differ in their cell wall structure and their molecules. But remember, the archaea are not bacteria. They are their own domain. The bacteria which have different cell walls include the mycoplasma. And as I mentioned earlier, the mycoplasmas lack a cell wall. And that's why the cells are pleomorphic. They don't have a cell wall to give the cell a shape. So the cell can take on essentially any shape. The mycoplasmas may have sterols in the plasma membrane to help give the plasma membrane strength. And as you recall, humans or animals have sterols in their plasma membrane to help give the plasma membrane strength. For us, the main sterol is cholesterol. All right, the last bacteria which have different cell walls are the mycobacterium. We will talk a little bit about them later. They are a gram-positive cell, but they're different because they have a lot of lipids in their cell wall. And the mycobacterium make mycolic acid and then put that mycolic acid in their cell wall. That gives the mycobacterium different properties from other of the gram-positive bacteria. Mainly, the gram stain doesn't stain well in the mycobacterium because the gram stain is aqueous, it dissolves in water. And the mycobacterium have so much lipid in their cell wall that the gram stain does not easily get in the mycobacterium. It usually does get in, but it'll stain lighter than the gram-positive cells. The mycobacterium have acid-fast staining properties, and that's because if you treat them with an acid dye, the acid gets through the lipid and then will stain the cells. Whereas other bacteria that don't have mycolic acid in it, the acid fast probably does get in the cell, but it then washes off in the decolorizing step. And the acid stain does not wash out of mycobacterium in the decolorizing step because the cell wall helps keep that acid fast stain inside it. All right, any questions about the irregular bacteria? If not, this is a good place to stop. So I think I'm going to stop here. And then I'll see you at 6:30 in the lab.